So I don't forget, I first want to mention this was the first year for lunchtime workshops and the liver sessions. One covered fatty liver disease that was coordinated at lunch, that was covered by Desley Neal, and it showed that we have a terrible problem storing fat in livers. So she's planning to coordinate a set of rules by which to score fat, and it's done differently all throughout the world. So we had cases that were scored from 5% to 90% on the same biopsy, which is not so great. But we look forward to hear more from her next Banff meeting. And we also had a workshop on digital pathology, which was headed by Brad Ferris and Drew Lesniak. It went over very well. There was a discussion about techniques and availability. The outcome of the meeting was that we're going to try to create a database of people that are writing algorithms, people that have particular staining capabilities or analytic capabilities, so that the wheel isn't reinvented at each and every place. The goals of the 2019 liver meetings, we had 8 presentations over a period of 2 days. We wanted to correlate histopathology with molecular profiling, and Alberto Sanchez Fuego is heading that effort with the goal of ultimately integrating molecular signatures for different types of allograft dysfunction into the Banff schema and correlate that with histopathology. We were particularly interested on why certain genes are elevated under certain circumstances, and can we iterate between the tissue and the gene arrays to actually work out details of the pathophysiology. And we focused on the major problem in liver transplantation, which is the minor infiltrates with low-grade interface activity in long-surviving liver allografts. And the goal was to use multiple modalities and come to some consilience on solution of problems to move forward. So we tried to incorporate serology with HLA typing and DSA analysis, the molecular phenotype, and routine and more sophisticated histopathology. The session was started off in an elegant fashion, as usual, by Stefan Hupscher, who really set the stage and described the extent of the problem, which is significant. If you look at long-term liver allograft survivals, and some of the figures are shown up on the screen there, more than half of the patients by 10 years after transplantation have variable degrees of chronic inflammation with interface activity and fibrosis. It's very concerning.
Maximize the image.
Yes.
Oh.
Just maximize.
I did not see your slide. Oh, that's better.
Better? So you can see the time post-transplant and the abnormal histology, the majority. It's worse of a problem in pediatric patients, probably because of compliance, but also there's a questionable value equation in adults for treating this with increased immunosuppression, partly because people didn't know what it was until a few years ago, but now they still are not decisive on how to treat it. So the commonest histopathologic findings are inflammation and fibrosis that increase in time, and there's increasing recognition and pretty good proof now that it's immune mediated, and in some cases antibodies contribute to that injury. So what we did in the intervening 2 years, we did a study with I have to be careful of this name. The full name is actually Gigolo. It's a graft injury group study, and we looked at the reliability of findings that may help us recognize chronic antibody-mediated injury. These are a heat map showing the weighted kappa values here. You can see the agreement. When it's green, it's very good. The kappa value is greater than like 0.8. Yellow, it gets a little worse. Red, we didn't have too much red. There were some features that were described in chronic AMR that weren't reliably identified by a group, and this was 6 pathologists across the United States, Europe, and Asia. So some of those findings were dropped from the assessment. His summary conclusion was abnormalities are frequent in late post-transplant biopsies, even when there's excellent liver function tests. Most common findings are inflammation and fibrosis, and most cases of this probably represent alloimmune injury and involves probably both T cells and antibody-mediated injury, but further studies are needed. The next presentation, we flipped to the molecular side for very elegant studies done by Richard Taubert from Germany, and he combines kind of fancy immunohistochemistry with multiplex labeling and molecular profiling on protocol biopsies. He looked at this chronic injury problem, and you can see there is a gene set associated with it. The markers are shown up here. I believe he used the molecular microscope. On the left panel, you can see that it's no histologic rejection, NHR. The middle group is this subclinical TCMR, which is this minor infiltrate, and then the right-hand side of the heat map shows you the genes that are upregulated in rejection when it's clinical, the TCMR, and it increases gradually as you go from left to right. He then tried to look at DSA positive patients with some features of chronic AMR, and you can see the Venn diagram up in the upper left hand side of the image. The genes associated with clinical TCMR or chronic TCMR and chronic ABMR, 50% T cell immunity, 50% inflammation pathways, and tissue damage. The group with non-overlapping chronic ABMR had the same pathways as the overlapping group, and genes unique to chronic TCMR were cell cycle activation. The important point here, which is also shown later on, that there was no absolutely discriminatory genes for chronic AMR, like you might see in the kidney with NK cell transcripts and endothelial cell signatures, and we discussed many reasons why that might be possible. And he described his plans about moving forward using single-cell RNA analysis and then high-dimensional plotting with the TSNE plots, that they're just starting on that and using the human protein atlas. So his conclusions: on a molecular level, subclinical TCMR is nearly indistinguishable from biopsies without histologic signs of rejection. Both TCMR, chronic TCMR and chronic ABMR are characterized by T cell signal pathways. Possible chronic ABMR, because it's still work in progress in the liver, lacks specific B cell molecular pathways, and he observed only minimal overlap when he looked at the 341 chronic TCMR specific transcripts and the published 13 TCMR specific transcripts that were determined by microarray from another group. The next elegant presentation was by a very experienced clinician, Jan Leroux, and he discussed how the clinicians react to biopsy findings and immunosuppression, and his particular view on the field of transplantation, which the audience greatly enjoyed and encouraged him to write this up for an article. And he looked at the evolution of immunosuppressive agents, and pointed out that more recently there's a plethora of drugs that are available, and many patients at many centers are on corporate-sponsored clinical trials. So the approach to immunosuppression in many centers is not uniform. They're on many different drugs, and he felt that that was inhibiting the development of transplantation in some respects. And he said, you know, all drugs have different side effects. There's too many choices and too many different schemes, so it makes it harder to interpret data. And despite all these new drugs, as is true in kidney and other organs, even though we've tried to really stamp out acute rejection to an almost zero incidence, the incidence of long-term graft survival really hasn't improved much, and that has been true for liver to the same extent it has been for many other organs. And Jan has, for 20 years, been very tightly titrating the immunosuppression, and his goal is to advocate for a shift in immunosuppression to go to the lowest level of immunosuppression that's possible without risking allograft failure, so that you can avoid renal and neurologic toxicity and infectious complications. And it's particularly true in adults because more adults die of immunosuppressive complications, but this puts it into perspective for the pathologist when he's diagnosing these minor infiltrates and what you do, how you react to them. So he felt that there was a need to rethink the concept of what is standard immunosuppression. The multi-drug regimens he feels are not always well thought out and can be somewhat expensive, and which was good news to the group. He's been a very strong advocate of pathology with protocol biopsies and puts the biopsy findings together with the clinical findings. The next presenter went back to the molecular profiling. He wasn't transplant specific, but he was liver specific. And one of the aims of the meeting was to correlate morphology with molecular profiling. So he was looking at risk factors for the development of carcinoma in patients with chronic liver disease, and found that the molecular profiling could actually pick up ectopic lymphoid structures, which is a risk factor for the development of carcinoma in the tissue. And you can see how he correlated it with the histopathology, with the present of pleural tracts with ectopic lymphoid structures, and you can see the heat map signaling that identified that in the tissue. This was also an elegant study that he went through the molecular profiling of animal models for fibrosis development in the liver. You can use diet, you can use carbon tetrachloride treatment, bile duct ligation. And the reason they went through all these different species because they wanted to determine whether the molecular profile of the development of fibrosis was similar to the human, which I think is shown on the far left column. So that they studied appropriate animal models to both reverse fibrosis and to prevent development of hepatocellular carcinoma. His conclusions was demographics that involve cell types can be monitored by the molecular profile. The presence of histologic features can be detected. Landscape of dysregulated molecular pathways allow unbiased, hypothesis-free exploration of therapeutic targets. And he illustrated a couple of those therapeutic targets in animal models where he could prevent the development of hepatocellular carcinoma. The second part of his presentation was molecular profiling in liver disease combined with clinical parameters can improve prognostic scores for patients. This was done particularly on acute and chronic severe alcoholic hepatitis. This is a controversial area in liver transplantation. Should you offer transplantation to somebody that has superimposed acute alcoholic hepatitis on top of cirrhosis? And he thought that molecular profiling might be able to better predict good prognosis patients from bad prognosis patients. So we looked at the cohorts, and they had a kind of a gene signature altered MELD score, and that was the best. So if you take the regular MELD score, which is the model for end-stage liver disease, and layer on top of it the genetic profile, you can greatly improve the prognostic ability to determine who might benefit from transplantation. The next presenter was Chris Bellamy, who gave a very elegant presentation and a very deep dive on a lesion that's been around for as long as people have been looking at livers under a microscope, and that's interface hepatitis. And there's very few deep dives on this lesion to look at the microenvironment of what's happening at this location. The reason this is important to study is it has shown up in every chronic liver disease, chronic hepatitis C, hepatitis B, autoimmune hepatitis. It's a well-documented prognostic indicator of progression to fibrosis and cirrhosis, but we don't know what it is. Here's the lesion here, where the lymphocytes go into the edge of the lobule. And he looked at the microenvironment. I'm not going to go into all these details, but he looked at every cell in the microenvironment and how it might be contributing to this reaction and why it might be an anatomic location or a nodal location of how the liver processes and distributes immune response. And he came up with this very detailed diagram that also the group encouraged him to publish and write this up, because people really haven't studied this in any detail. This does not exist in the literature where you look and say, okay, what is the microenvironment of interface hepatitis? The next presentation was by Valeria Moss from University of Tennessee. She looked at molecular profiling of rejection and tolerance across different organs. She pointed out the pros and cons of the different approaches and the different platforms, and really highlighted on this slide the problems with batch variation and how there are emerging software toolkits now to deal with these batch variations that will make inter-lab analysis and collaborations more common. But she said it is a significant problem in the gene array field. Her overall conclusions is that these newer technological and computational advances will allow better data integration and meta-analysis. Study size for tolerance studies are often limited, and they lack proper control, so she felt that some of the gene profiles that are generated may not hold up the test on independent validation cohorts. Jackie O'Leary gave a nice presentation on morphologic chronic characterization of chronic liver allograft AMR and how it might be improved. But before that, she showed a case series of simultaneous liver kidney allograft recipients from the same donor and sensitized recipients, where you get timed biopsies from each allograft with simultaneous DSA determination. This is just one value, and I think it might inform some of the correlations between morphology and mRNA expression arrays. On the top row is a kidney allograft. This is the sensitization profile class 1 with an MFI of 6,700. Kidney biopsy on day 15, liver biopsy on the bottom. At day 13, C4D stain is on the far right column. You can see the capillaries in the kidney are diffusely positive. Look at the liver, only the capillaries in the portal tract. So when you do molecular profiling, the vast majority of cells that are identified transcriptionally as endothelial cells don't have C4D deposits in it. But that doesn't mean the liver isn't experienced AMR, because you can see the same pathology in the liver as you see in the kidney up here. you get the capillaritis and endothelial cell damage. So whether it's a sensitivity issue or whether nanostrength might be able to tease this out so you have better correlation between the morphology and the gene expression profile, or works in progress. Her conclusion was chronic AMR does occur. It's unclear if the injury is mediated directly by antibodies or the antibodies facilitate cell injury, or both, probably both in the majority opinion. Some histopathologic characteristics associated with chronic AMR, this interface hepatitis, portal venopathy, fibrosis, central vein, and periportal. She felt that there was a need for more precise histopathological criteria, and that will be worked on in the interval between the next meeting. Stress the importance of protocol liver biopsies. If you don't do a protocol liver biopsy, you don't know that many patients have fibrosis and chronic hepatitis because the liver function tests are normal. And reminded people of how the liver is different in terms of the two-hit hypothesis, which is important. You have to have perturbation in the liver to upregulate the class 2 targets on the endothelium. And she suggested a plan to do a randomized trial in adults with to address this lesion in a larger patient cohort. And then finally, the guy that's going to get this all together, I don't know if it'll be after this meeting or at the next Banff meeting, to correlate the histology with the molecular profiling and give some consensus with respect to the liver. Talked about rejection Dsa, and again, this pathogenesis of subclinical liver damage. Here you can see the transcripts of ABMR in other organs and TCMR, but what's important here is to point out the molecular profile of the genes also overlap in other organs. So a lot of the genes are shared, which is probably true, and it reflects the pathology, at least in my experience. Whether it's heart, liver, kidney, most of the time I see a mixed rejection picture. It's, in my experience, uncommon to see an absolutely pure form. You can see it, but it's not that common, and I think he felt that that was one of the reasons that the molecular profile overlaps as well. He did all the molecular analysis for Sandy Fang study, and he tested his 11 gene rejection signature in patients from cluster 1, which was a group of patients with this interface activity, versus a normal biopsy, and you can see his T cell or his molecular set of genes nicely predicted and were upregulated in patients with this low-grade interface activity compared to biopsies that were normal. Then he looked at genes that might affect the tissue, sorry about that. He looked at whether circulating DSA influences the intrahepatic gene expression profile. So he looked at ABMR selective genes from kidney transplant, TCMR from kidney, and TCMR from liver, and basically really wasn't able to tell the difference, and they overlap. So what he did was a normalizing technique using our multiplexed immunohistochemistry. So he normalized it with respect to the load of CD45 infiltrate over the biopsy, and he was able to show that when you have circulating class 2 DSA, that CXCL9 and CCL18 were independently associated with DSA. So we're going back into the tissue and trying to look to see where that might be produced in the liver to help us better understand the processes. So in conclusion, from his study, it showed that biopsies with inflammation and interface activity shows a transcriptional profile that closely resembles TCR and correlates with the magnitude of fibrosis. Biopsies of fibrosis but devoid of inflammation exhibit a distinct molecular profile suggesting non-immune injury. With the exception of the gamma interferon response, the transcriptional changes associated with ABMR in kidney and heart, which are the NK and endothelial cell signatures, are not really detectable. And we had a long discussion about that, whether it was a sensitivity effect, whether it was the antibodies were facilitating the T cells, whether we needed to do more targeted microdissection of the tissue, like the digital spatial profiling, or whether the liver gets masked, the signal gets masked in the liver. For example, the liver has a lot more NK cells naturally and normally, and you might not have to recruit them from the periphery. They might just relocalize in the liver, but it's the largest repository of NK cells in the body. And finally, he showed that the presence of circulating anti-class II DSA has a small independent impact on the liver transcriptome. So thank you.
He's not taking any questions. Are there any questions from the audience?
Okay.
Sorry, just one. Do we currently recommend testing for C4D in the liver biopsies?
We do it routinely, and in the absence of any routine DSA testing, I would probably default to an answer of yes. It's cheaper than doing the complete DSA testing, but otherwise, you don't have any tool at most centers to determine whether somebody popped a de novo DSA after transplantation. Is it an efficient way of doing it? Probably not. If the C4D is at least focally positive, in our experience, probably over 80% of them have a DSA. But I would say it's a rare lab or non-existent lab that at least does one protocol DSA, say a year or two after transplantation. So you don't know who this low-grade activity is going on. At our center, we do use the biopsy to trigger DSA testing, and when we see at least focal positivity, sometimes it comes up diffuse, almost invariably they have DSA.
So